Welcome to Chapter 5 on microbial metabolism. In this chapter, we'll be moving beyond the basics of microscopy. There's a limited amount of information that we can gain from looking at various gram stains and analyzing the cell wall structure, arrangement, various shapes and sizes of bacteria. So, in order to make a genus and species identification, sometimes one of the most efficient ways to do so is to look at the lifestyles of bacteria biochemically. How do they make their living? And pictured here are some E. coli that are computer colorized. But this is an actual picture of E. coli cells inside of the intestine, and they are attached to microvilli. And one thing we cannot tell from this microscopic picture is whether or not these organisms are healthy E. coli that are considered normal flora, or if in fact maybe they are a dangerous strain that could cause severe human illness or even death. And so one of the deadly strains of E. coli that causes severe disease is E. coli O157H7. And so strains of O157, which the O refers to an antigen that's found in the outer membrane of E. coli. This specific 157 antigen denotes a type of E. coli that, when grown on sorbitol, supplemented into an agar called McConkey's agar, which we use ordinary McConkey's agar in our micro lab. But then if you supplement in this sugar alcohol called sorbitol, then the medium is abbreviated SMAC. And E. coli O157 is known to be a non-fermenter of sorbitol. And this is an important fact because since most strains of dangerous E. coli in the O157 category are non-fermenters, they will appear colorless on the McConkey's agar, the SMAC agar. And this is in contrast to the normal flora beneficial E. coli that all humans carry that are generally sorbitol fermenters and turn dark pink or red on the medium. So in less than 24 hours, or I should say an overnight 24-hour culture, when you come back and you look at the plate, if you see clear colonies from a person's clinical sample, whether it be a stool sample or a blood sample, and these clear colonies result, this would be considered a suspected positive, a presumptive positive result for this dangerous strain of E. coli. And this is advantageous mostly because of the speed of this type of technique. Genetic analyses may be less convenient or more expensive, maybe more time consuming to perform, especially when looking specifically for antigens such as O157. And this is a very simple way in which the bacteria can be screened quickly. Metabolism as an overall process within an organism consists of anabolism and catabolism added together. So anabolism is a process by which an organism is building or synthesizing key molecules or components within the cell, whereas catabolism, by contrast, would be the breakdown or the breaking of components in order to release energy or to make use of those items in a different way. And overall, together, they make up the complete metabolism of a given organism. For clarity, here are the definitions of catabolism and anabolism provided. Both anabolism and catabolism are reactions that are connected to one another, so we can see them interconnected by a circular set of events. Beginning at the top of this diagram, We see simple molecules, building blocks, glucose, amino acids, glycerol, and fatty acids, as individual components that through the process of anabolism can be used to build larger, more complex macromolecules. And so then glucose can give rise to starch, and amino acids can give rise to proteins. And we know that glycerol and fatty acids make up triglycerides, which are part of the basic lipid structures that we know of as macromolecules. These anabolic reactions are never 100% efficient, and they do require energy in the form of ATP to be hydrolyzed and used to build these buildings, to build these structures. But because it's not completely efficient, some of the energy that's used in this process will be released as heat. So all organisms will release heat during the course of their metabolism because of inefficiencies as energy is lost to the environment. Likewise, on the other side of this circle, sometimes an organism will have to take these complex macromolecules and break them down, possibly to get energy, to release energy. So if an organism is burning starch or proteins or lipids and breaking them apart, we can see that that's in an effort to make ATP to get more energy. But during this catabolism, it is not 100% efficient. So not all the energy that's released will go towards making ATP. Some of that energy will be released as heat. So whether an organism is undergoing anabolism, anabolic type reactions, or catabolic type reactions, some amount of heat energy will be released. Also, it's important to keep in mind that during anabolism, which is the pathway of taking building blocks and making complex macromolecules, ATP is actually being used during this process. It takes energy in the form of ATP to build these structures. It takes energy to build a building. So ATP is being used even though we're talking about an anabolic pathway where things are being made. And on the other side of this event, when an organism has needs to make more ATP, they actually have to break apart some of these valued structures, whether it's starch, proteins, or lipids, or even nucleic acids for that matter. When an organism breaks down those macromolecules, it's in an effort to cash out and gain some ATP, to make some ATP. But keep in mind, even though ATP is being made here, this is during what would be considered a catabolic form of metabolism. So when we say anabolic, we're referring to what is being made in terms of macromolecules. When we say catabolic, we're referring to what is being broken down as far as macromolecules go. Metabolic pathways are driven by a series of enzymatically catalyzed reactions. And the goal of most of those reactions is to create a way to store or release energy. The big E in this slide is referring to energy. And there are various shapes or patterns that these pathways can take on. One of the most common would be a linear pathway, where molecules can move, say, A to B to C to D. And one common example that pops up in my mind is presented later in this lecture, intermediate step. The intermediate step occurs during aerobic respiration, and it precedes the Krebs cycle. Some pathways do not take on a fully straight set of steps, and they can actually be branched instead. So a fork in the road may occur where a molecule could take one or more paths. An example of that would be pyruvate metabolism. So pyruvic acid or pyruvate is a molecule that can go in different directions based on prevailing conditions within the cell. And so we'll see how pyruvate can be part of the beginning of a branched metabolic pathway. And finally, there can be circular pathways, like the Krebs cycle that was mentioned as an event that is cyclical and happens after the intermediate step during respiration. Ultimately, metabolic pathways are determined by enzymes, and these enzymes cannot exist without the genes in the DNA that encode these enzymes, which are made of protein products. And so it's all interconnected here and driven by the enzymes that make these pathways possible. When we look at metabolism as an overall process or in concept, we could ask the cosmic question, why do we need metabolism at all? And the answer is that metabolism provides any organism, all organisms, with the ability to store and release energy appropriately. If we didn't have a way to store and release our energy appropriately, then we would have to take in food pretty much minute to minute before each and every move that we would make. And so metabolism is key in allowing us then to make use of foods, most often carbohydrates, and those provide then most of our cellular energy. And so glucose, being the most basic carbohydrate, is the most common. It makes up our blood sugar, and most organisms prefer glucose. Now, don't be confused. Lipids are the most energy dense. So per gram, lipids will provide more energy, but they are not the most common source of energy for most organisms. So we prefer sugars, but lipids still contain the most energy. In order for any chemical reaction to occur, the chemicals that react must come into close contact with one another. And this can be defined by collision theory. And essentially, collision theory states that molecules or atoms must make physical contact for a reaction to occur. Chemical reactions can involve the formation of bonds that result from the rearrangement of valence electrons that will make these bonds possible. And so collision theory is important for there to be enough proximity for these electrons to rearrange. Also associated with the chemical reaction must be a sufficient amount of energy to make this possible. We call this activation energy. It's defined as the amount of energy required for a chemical reaction to occur. And below this energy, the reaction may fall short of occurring. And there are ways to manipulate this. In the chemistry lab, we can do things like heat up a reaction or stir it up along with heating. But there have to be ways to meet that sufficient amount of activation energy. And the rate at which products form is referred to as the reaction rate, or the speed of a reaction. And if you were to put this onto a clock or a time scale, we would say that the speed of a reaction is measured in the number of products that are formed per second. One way to think about collision theory is to think about how chemistry lab experiments are conducted. Oftentimes, students have a laboratory setup with various equipment that involves facilitating chemical reactions by heating. So we can increase the temperature in a given environment to increase collisions of molecules, so they run into each other more often. Temperature is actually the measure of molecular motion within a given system. We could also pressurize a system. So if you create pressure, that'll cause molecules within a given reaction vessel or system to collide more often. Also in the chemistry lab, you might throw in more chemicals and increase the concentration. So if there are a higher concentration of chemicals, those chemicals that may react will run into each other more often. We could also manipulate the pH. Occasionally, chemical reactions can be inhibited or increased by the appropriate pH in a given reaction that's being studied. And stirring, also referred to as agitation or shaking, is another way that we can increase the amount of molecular interaction or collision. So these are some of the common ways in which a chemistry lab setup might manipulate the system for this reaction to occur according to collision theory. Unfortunately and inconveniently for the human body, these are not options that we can exercise to any great extent. There are limits to how much we can increase our body's temperature. We have a narrow range at which we can function without harming ourselves with too high of a temperature. We can't really internally pressurize ourselves, and likewise we can only eat so much food or increase the concentration of chemicals necessary for reactions to occur. So concentration is limited. pH does sometimes play a role in some enzymes and where they function, but generally speaking we can say that the human body maintains a fairly neutral pH in most organs and tissues, so pH changes are limited. And agitation. Unless you want to jump around and shake yourself up, there's only a limited amount of motion you can create through agitation. Therefore, with all of the limitations that the human body has, we employ the use of one other tool that's not quite as common in basic intro chemistry labs, but we can add a catalyst. So occasionally chemical reactions are facilitated by catalysts, and a catalyst in the human body would be an enzyme. Enzymes are able to facilitate a chemical reaction without the need for dramatic increases in temperature, pressure, concentration, pH, or agitation. Catalysts do the work in the form of enzymes, and those enzymes are able to bring molecules or atoms together and cause collisions to occur most efficiently. And it decreases the amount of activation energy required for the reaction to occur. This graph shows the energy requirements for a chemical reaction with or without an enzyme. So here we have a molecule or reactant that is labeled AB, and so let's imagine by example that could be a molecule of starch. So you have starch, a polysaccharide made up of many glucose molecules, and then imagine if you wanted to break down AB as starch into separate molecules A and B. And so maybe it would be 2 molecules of glucose at the end that have been broken apart. And the first line on this graph, the taller one, or the taller hill here, shows how much energy would be necessary to break apart that starch without any enzymatic catalyst, like amylase, for instance. So without an enzyme like amylase to break down starch, the amount of energy necessary could be nearing boiling temperatures. Then if you were to put in some amylase, we would see that there would be a dramatic decrease in the amount of energy that would be necessary to cause the breakdown of starch into its separate glucose end products. So let's do some labeling here. Without an enzyme, boiling temperatures may be necessary. But add in something like amylase, and now this same reaction could be carried out efficiently at body temperature. And that's a dramatic difference in the amount of energy that's required by the system. Boiling is nearly 100 degrees Celsius, or I should say is 100 degrees Celsius, whereas human body temp is only 37 degrees Celsius. And the one difference between boiling and human body temperature that makes all the difference is the amylase enzyme. So the take-home point here is that enzymes decrease the activation energy required for a chemical reaction. Enzymes have a three-dimensional globular protein structure that is precisely configured to complement the substrates that they bind and interact with. And enzymes have a technical name that ends in ACE, so the suffix ASE is always attached to the technical name of an enzyme. And we can say that enzymes are designed to catalyze chemical reactions by lowering how much activation energy is required. And the enzyme has a specific site on it called the active site, and that is the binding site that's specific for the substrate that it interacts with and then performs a biochemical reaction on. And we say that each enzyme catalyzes only one reaction. And these are basic rules that we can apply to enzymes as a beginning understanding. There are some exceptions to these rules that have been laid out. For instance, some enzymes are not protein-based and can be RNA-based, but we can say that the vast majority of enzymes do have a protein structure. And the bottom rule down here, each enzyme catalyzes only one reaction, that is true almost all the time. And there are some exceptions to the rule, and we can talk about those later, like beta-galactosidase is an enzyme that can perform more than one reaction, but that is something that's a relatively rare exception. And so one type of reaction is what we'll focus on at the moment. The molecular anatomy of an enzyme involves many components, and we begin with the most important or largest component of the enzyme, which is the apoenzyme or protein portion of an enzyme. So almost all enzymes have this apoenzyme protein portion. And many enzymes also have a non-protein portion referred to as the cofactor. And together, the cofactor and the apoenzyme will make up a fully functioning enzyme. And cofactors can vary in what they are as non-protein portions. Some cofactors are inorganic minerals, so we can think of trace elements such as iron or magnesium or manganese or something of that sort. Or you can have vitamins, organic coenzymes. So think of the letters of the alphabet: vitamin A, vitamin E, vitamin C, etc. And so you can have these cofactors be either organic or inorganic. And together, the cofactor, regardless of which type it is, and combined with the apoenzyme protein portion, we get the fully functional holoenzyme, as it's technically called. The holoenzyme, as the name sounds like, is a whole functional enzyme. And it'll be capable then of facilitating its specific chemical reaction that it's designed to perform. And holoenzymes may not necessarily be active at every moment, but they are fully assembled and can perform their activities because they contain both the apoenzyme and the cofactor. And then there are other events that can turn that holoenzyme on or off. And we'll be seeing examples of the on or off state of an enzyme or holoenzyme in some future slides and examples. The active site we know is the part of the enzyme where the substrate can bind, very specifically. And then there is what's called the allosteric site. The allosteric site refers to allo meaning other. And so the allosteric site is that space where it may act like an on-off switch for an enzyme. We can label it as a place that may turn the enzyme on or off, active or inactive. This is a picture taken from our textbook that illustrates the components of the previous slide. So visually we can see what an apoenzyme might look like with having a complex three-dimensional globular structure, and then combined with its coenzyme or cofactor, these two items can fit together to make the so-called holoenzyme. And then now this will have the properly shaped active site that can make contact with and bind to the substrate so that it can convert that substrate and perform a chemical reaction. Here's a list of important coenzymes that are found in various living cells. NAD, which carries a plus charge, is an abbreviation for nicotinic adenine dinucleotide. And similarly, there's NADP, which stands for nicotinic adenine dinucleotide with a phosphorus added to it, and also carries a plus charge. These, instead of memorizing the names of these, I'd like you to make sure and memorize the function, along with FAD as well, which is flavin adenine dinucleotide. All three of these are what are called electron-carrying coenzymes. So we'll label these as electron carrying molecules, and their complex organic structure has the function of picking up hydrogens and their associated electrons to shuttle them to other places in the cell. For varying biochemical reactions, ranging from respiration to hydrogenation, or a place where electrons and hydrogens are needed. And last on the list here, we have coenzyme A. And coenzyme A, which is sometimes abbreviated CoA, is involved in preparing a molecule that feeds into the Krebs cycle, as we'll see later. So these are common coenzymes that we should keep in mind as we go through this lecture. The speed and specificity by which an enzyme functions is surprisingly fast. Enzymes can have what's called a turnover number that refers to the reaction rate, and enzymes make cells extremely efficient in terms of how quickly these reaction rates or turnover numbers can be produced. Some enzymes can be on the slower side of generating maybe 1 molecule per second, but then there are many enzymes that can generate hundreds or even thousands, upwards of 10,000 according to our textbook, of molecules per second. And this is blindingly fast. It's incredible to try to imagine thousands of products being generated by the activity of one enzyme. In fact, beyond our textbook's description of this, a physiology professor in our department at OCC had mentioned that there's an enzyme called carbonic anhydrase found in human physiological reactions that can generate up to 1 million molecules per second in terms of its turnover number. So reaction rates can be extremely fast and difficult to conceptualize, but nevertheless, these are the numbers that have come out of enzymology, the study of enzymes and how efficient they are at turning over chemical reactions. We can say that the normal sequence of events for an enzyme follows that the substrate must make physical contact with the enzyme. And we know that the enzyme has a specific place for that physical contact, called the active site. So this would be step 1 for an enzyme to do its activity. And once the substrate makes contact with the active site, there is a temporary enzyme plus substrate E plus S complex that will form. And after a brief enzyme substrate complex, there will then be a biochemical reaction that takes place where we can state that the substrate is transformed and released. If we think about steps 1 through 3, it seems that the process would be complete. But I'd like to add on a 4th step to remind everyone that a major concept that comes with understanding enzymes is that all enzymes are recyclable, or at least their function is not to become part of the chemical reaction or their end products, but ultimately to remain unchanged and available to facilitate another chemical reaction, so that they can bring about this same chemical reaction over and over, and they do not become a part of the products or reactants. They only facilitate that the reactants will undergo the reaction to become the products. So we say that enzymes are recyclable. Here's an illustration visually showing the events from the previous slide. And although I outlined 4 steps that are part of the normal sequence of enzymatic reaction, we see that there are 1, 2, 3, 4, 5 events labeled from our textbook. We can still think of it though as 4 major events and make it a little bit simpler. And so here we can see the globular 3-dimensional structure of the enzyme, and the active site is this area on the top where the substrate can fit into the enzyme. And here we see the temporary enzyme plus substrate complex that forms. And then we see that the substrate is transformed and ultimately released. So step 3 and 4 are referred to in the previous slide as 1 step, that the substrate is transformed and released. And ultimately then we have an enzyme that remains unchanged and available to produce this reaction over and over again. Here's a picture showing another angle of this, in which we'll describe the induced fit model. And this is part of what occurs during the formation of the enzyme substrate complex. So on the left, we can see the substrate, and then as it fits into the active site of this globular enzymatic structure, what will happen is like a catcher's mitt, we can see the enzyme will flex and form this enzyme substrate complex as an induced fit. And then during this process, the substrate will be transformed and eventually released. There are 6 different ways in which enzymes can be classified according to their function. The first are oxidoreductases. And so oxidoreductases move electrons around, and one particular example that we've looked at in lab would be the nitrate reduction experiment. So nitrate reductase is a type of oxidoreductase. When the electrons are moved from one place, you get an oxidation, and where those electrons end up, you get a reduction, if you remember back to oil rig as an example. Transferases are yet a different class, where instead of observing that electrons are transferred, you will see that full functional groups are transferred. So we studied lysine decarboxylase and ornithine decarboxylase, and we also studied phenylalanine deaminase, and those would be examples of transferases that move around functional groups. Hydrolases have also been studied, and so we looked at the starch hydrolysis experiment, or ureases actually perform something called urea hydrolysis. And so hydro refers to water, and lysis means to break apart. And some enzymes require a water molecule in order to perform their digestive processes. Lyases are yet another class of enzymes that are referred to as, or described as, removing atoms without hydrolysis. And then there are isomerases that do a rearrangement or a reconfiguration of molecules, or the atoms within the molecule, without changing their molecular formula, but just rearranging them. And then Lastly, we have what are called ligases, where they ligate or join together molecules, and they are classified as requiring ATP energy. So a molecule of ATP would be used as bonds form between molecules and they join. There are 4 major factors that can affect the activity of an enzyme within a living system. And so we'll go through each one of these slide by slide. For now, though, the master list starts with temperature, and then we'll move on to pH, and then substrate concentration, and finally inhibitors. Regardless of which factors influence an enzyme's activity, we first must think about the enzyme as having an active, native, functional protein form. And so there are primary, secondary, tertiary, and even quaternary levels of structure, and they're delicately put together. So here we can see some alpha helices and some beta pleated sheets and an overall 3-dimensional tertiary structure. And then on the right, we can see the picture of a denatured protein. And one thing to take note of here is we see an unraveling. So the tertiary and secondary sequences are broken up, and the primary sequence still remains here as a big long ribbon of amino acids. And this denatured protein could actually renature if the damaging environment that is causing the protein to denature is repaired. This slide can be entitled Enzyme Denaturation, as the pictures show. But so long as the primary sequence does not get cut up in the denatured protein, it may be reversed. So some denatured proteins may be renatured if the proper conditions are restored. But only if the primary sequence is uncut or undamaged. So the proper pH must be restored, proper amounts of salt or other aspects to the environment that the protein is in, and perhaps then an active functional protein could reform. One of the most important factors on enzymatic activity would be temperature. Temperature is defined by the amount of molecular motion within a system. So at lower temperatures, the molecular motion is very slow, and it will take longer for enzymes to make contact with their substrates as the substrates move around at a slower rate. As we increase temperature on up the line here, we can see an increase in enzymatic activity because there's an increase in molecular motion, and more often there will be contact with the active sites of the enzymes. And this will continue to increase and increase with molecular motion up to about 37 degrees Celsius, at least according to this graph. So most enzymes work maximally well at 37 degrees Celsius, and then as our body runs a fever, our bodies will then experience enzyme denaturation, where rapidly we'll see enzymatic activity drop off. Most enzymes that we'll study physiologically and that are present in medically important bacteria will function best at 37 degrees. Beyond that, the drop-off in enzymatic activity can be explained by what's called denaturation. So the enzymes will lose their 3-dimensional structure and start spreading out into a non-functional conformation that can be described simply as denaturation, and then we will see that the chemical reaction will no longer take place as the heat goes well beyond the point of maximal function, which is typically at 37. Another major factor that can have a dramatic influence on enzyme activity is pH. And in this figure, the ideal pH, or optimal pH for an enzyme studied was right over 5. And so this must be a type of enzyme that's slightly acidophilic. Recall that a pH of 7 is neutral. And it should be noted that despite this graph, most enzymes function optimally at 7, as most living systems prefer a neutral pH. So most organisms are neutrophiles. The human body maintains a neutral pH in most organs and tissues. This could be a graph showing an enzyme that functions in the digestive system. We have stomach acid, and so some enzymes will work in a much lower pH than 7. And on either side of this peak, we could say that enzymatic activity drops off here and here on either side of the peak due to less than optimal pH for this enzyme. And so this would be denaturation once again. A place on the graph where the enzyme is not in its proper three-dimensional structure and therefore doesn't have the ability to do its function. One rate-limiting factor that affects how well an enzyme can do its job is how often it makes contact with a substrate. So if there's very little substrate within a system, then the enzyme won't have very much work to do. However, as the amount of substrate in a system is increased, we can say that the substrate will be worked on by various enzymes that are available. So we can see the activity of the enzyme go up and up and up very rapidly as we increase the amount of substrate that's available to react. Now, there is a point where fairly quickly, once the substrate reaches a certain concentration, that the enzyme activity will level off. And this particular leveling off effect is referred to as a saturation point. So when there's way more than enough substrate, we'll call this saturation. One way to envision this is to use an analogous example. I like to use monkeys and peanuts as an example. If monkeys, functionally speaking, are the enzymes within a system, and the substrate that the monkeys work on are peanuts, the monkey's hands would be the active site. They pick up the peanuts and they separate the peanuts from their shells. Now, if there are only a few peanuts around in the monkey's environment, then that reaction will happen only as quickly as the monkeys can pick up the peanuts. However, imagine if for a moment you pulled up a dump truck, a giant truck full of peanuts, and then dumped them into a room full of monkeys. And if all these monkeys or enzymes are completely surrounded up to their knees or their waists or something, just swimming in peanuts, then the reaction will hit a point of saturation where it'll level off because there's more than enough substrate to make contact where the shells are getting into the hands of the monkeys as quickly as the monkeys can shell them and move on to the next peanut and the next peanut. And so the enzymatic activity will level off at a point of saturation. The fourth and final factor that can affect enzymes is competitive inhibitors or enzyme inhibitors. So competitive inhibitors are specifically molecules that resemble the normal substrate. Okay, so back to the monkeys and peanuts analogy for a moment. If the monkeys are the enzyme and the substrate are peanuts, imagine for a moment if in that system you threw in plastic peanuts. So a plastic peanut looks like the normal peanut. It could fit into the active site, so it competes with the normal peanut for the monkey's hands. But then there's no way to separate the shell from the nut because it's something plastic. And so this will slow down the normal reaction that's intended. And the reason that competitive inhibitors are important to understand is that they can actually be used as a tool pharmaceutically. So there are drugs called sulfa drugs. Sulfanilamide specifically is a type of sulfa drug that resembles the plastic peanut in the sense that it looks a lot like a natural substrate. And instead of talking about peanuts for a moment here, we'll say the natural substrate is something called PABA. PABA stands for para-amino benzoic acid, PABA. And this PABA is essential for the synthesis of something called folic acid. And folic acid is a type of B vitamin. B vitamins, like folic acid, are involved in DNA synthesis. They are precursors to nucleotides that are key for DNA synthesis to eventually occur. And it's important to understand that the human does not make its own B vitamins, but some bacteria do. So if some medically important bacteria, such as E. coli or staph, typically can make their own B vitamins, like folic acid, then they can go on making their DNA and growing and multiplying. The human, however, must get their folic acid through dietary intake. So it's common, by example, for pregnant women to be given a folic acid supplement. And the reason for that is if the human body becomes deficient in folic acid, then we would have difficulty making our DNA because we can't make our own B vitamins. And if you don't get enough in your diet and you're growing a baby like an expectant mother, then that baby could have birth defects due to the fact that not enough DNA synthesizing building blocks would be necessary or would be there. And so B vitamin deficiency is very dangerous as it can bring about spina bifida or cleft palate and other neural tube defects that are related to vitamin B deficiency. But ultimately, we can say that the sulfanilamide, or so-called sulfa drug, is an inhibitor to bacteria, but not to the human, because humans are not able to make their own folic acid, but some bacteria are. The sulfa drug interferes with these bacteria by inhibiting the folic acid synthesis pathway. And like I said, that sulfa drugs can be envisioned as the so-called plastic peanut within the monkeys and peanuts scenario. If you flood the environment with sulfa drugs, then that bacterium will take the sulfa drug and pick it up, thinking that it's useful for making its folic acid. But instead it picks up the sulfa drug, which doesn't function to give rise to the folic acid. And so every time the bacterium picks up the sulfa drug, this slows them down. Their enzymes that make folic acid are fooled into picking up the sulfa drug. And so this blocks folic acid synthesis. And as a final note here, it should be important to understand that this type of effect, this type of drug, must be applied continuously and at a proper level so that the environment in the human body has enough of the sulfa drug that the bacteria are constantly being hit with something that they aren't able to make progress with. If the patient is instructed to take these sulfa drugs several times a day so as to keep the concentration up, then the drug will be effective. However, if the person does not take the drug, but maybe forgets, and then by the end of the day is behind by 2 or 3 pills, then the concentration of the sulfa drug, aka the plastic peanut, will fall, and during that time the bacteria will be able to make the progress because there isn't this interfering factor. So it's important to understand that with inhibitors, it says here that this type of inhibition can be reversed by increasing the concentration of the natural substrate, or alternatively by not taking the drug. Then the inhibitor won't work or compete if it's not at proper levels. Here's an illustration that visually shows what the previous slide describes in words. On the left, we have the normal substrate, like the natural peanut, making contact with the enzyme, which I had compared to the monkey's hands. And this is how folic acid could be produced under normal circumstances inside the bacterium. So the normal substrate is PABA, and that PABA, para-aminobenzoic acid, will then fit into the enzyme and eventually give rise to folic acid. However, the competitive inhibitor, which is the sulfa drug, is not exactly PABA, but is similar enough to the natural PABA that it can fit into the active site and then compete and block out the normal substrate's ability to contact the active site. And so this effectively slows down the number of times that the natural substrate can contact the active site. And so it can slow down or block folic acid production. For reference, here are the two molecules to compare. You can see their visual similarities. The sulfa drug, which is equivalent to the plastic peanut, is here on the left, and it has a benzene ring, and we can see this modified sulfate up here. There's a sulfur with some oxygens and an amino group, and so this is why it's called a sulfa drug. We see the presence of sulfur, and then here we can see the naturally occurring PABA, para-aminobenzoic acid. We see the benzene ring, we see the amino group down here, and then we see a carboxyl group up here. And we know that the naturally occurring PABA leads to folic acid synthesis. And humans don't have this pathway, but bacteria can make their folic acid, which allows them to make more DNA and grow and multiply. And we know that the sulfa drug will block this event from happening as it makes contact with the folic acid producing enzymes that need PABA but instead contact sulfa. Enzymes may also be inhibited by non-competitive inhibitors, and the main difference here is that a non-competitive inhibitor will not compete with the substrate for the enzyme's active site like competitive inhibitors would do. One mechanism of how non-competitive inhibitors can work is through what's called allosteric inhibition. And in a previous slide, allosteric was defined as an other space a non-active site that does not bind with the substrate. And when this non-active site or allosteric site is bound with a non-competitive inhibitor, it may change the shape of the enzyme so that the enzyme's active site can no longer make contact effectively with the substrate. Allosteric inhibition is something that can occur naturally within a cell and does not have to be an unusual event. It can be part of nature's evolved plan within a cell to turn an enzyme on or off. And the way that this works typically is that an end product that's made by an enzymatic pathway will build up, and when that end product builds up to a sufficient level, it can act as a regulatory mechanism where it binds to the allosteric site on the enzyme and temporarily shuts its activity down. And then when that end product's concentration falls, then the enzyme may then go back to its active state. Another form of non-competitive inhibition may occur through poisoning with heavy metals. And you may have heard that certain heavy metals are dangerous to various life forms, ranging from humans all the way down to bacteria. There are certain heavy metals that can get into the environment and into living cells and poison them. And the way that this poisoning usually works is through denaturation of enzymes that the heavy metals bind to or make contact with. Let's start by looking at a few normally occurring beneficial metals that are required by many organisms. So when we think of the human, we know that we need iron. If we're low on iron, we may become anemic because our hemoglobin requires iron. And iron can come in a +2 or +3 charge. And then magnesium is another essential trace element. And plants need magnesium, especially for structure of their chlorophyll, which is a complex enzymatic structure that depends upon the magnesium to be shaped properly. Zinc would be another common element that many organisms require some of for their enzymatic structure, as well as manganese. And so these are just some examples of normal beneficial metals. By comparison, though, toxic so-called heavy metals, which are often greater in their weight, so hence the word heavy, can refer to metals such as lead, which also has a +2 charge, and mercury, +2, and copper, and possibly even silver, as just some common examples of heavy metals that can go in, and the way that they are toxic is that they displace the normal natural metals that are in these enzymes, and when they bind to enzymes, their affinity for those enzymes may be greater than the normal metals, and so then they bind in a way that they will not easily unbind, and if those enzymes are taken over, they can change shape when they're in contact with these toxic heavy metals, and it can shut an organism's ability down in terms of just everyday biochemistry that these enzymes carry out. Here's an illustration that visually shows what the previous slide was communicating, where you have an enzyme with its three-dimensional globular structure, And the active site normally is shaped to make contact with the substrate and bring about a chemical reaction. However, in the second figure on the right here, we're seeing that if a non-competitive inhibitor enters the environment, it will have a shape that makes contact with an allosteric site. And when this binding occurs, it effectively changes the active site's three-dimensional shape so that the substrate can no longer make contact. And this type of inhibition then shuts down the enzyme's ability, at least temporarily, to do what it's designed to do. So whether that non-competitive inhibitor binds temporarily and then shuts us down and then unbinds at a later time to turn the enzyme's activity back on, or if a heavy metal binds in here and effectively changes the shape, which would then be a less reversible process. Either way, the key concept is that the non-competitive inhibitors are not competing or interfering directly with the active site. They are making contact at some other site or space on the enzyme's structure. Now that we've learned a bit about how non-competitive inhibition works, we can look at feedback or end product inhibition as a way that cells can use this form of inhibition as a negative feedback loop mechanism. Okay, so this slide is entitled Enzyme Inhibitors with Respect to Feedback Inhibition, otherwise known as a negative feedback loop physiologically. And the way that this works is an enzymatic pathway may move from A to B to C to D, but at the end of that, let's say D is the final end product. That end product, when the enzymatic pathway produces enough of it, will serve to inhibit the enzyme's activity. And so the way the end product does this is it binds allosterically to the allosteric site on the first enzyme of that pathway. So if we talk about A moving then to B, then moving to C and to D, we would be saying that conceptually D will loop back and bind allosterically to A on enzyme A's allosteric site. And then enzyme A's allosteric site will deform so that its active site can no longer bind to substrate. The pathway will shut down. But this is reversible because as the end product's concentration falls, as A to B to C to D is no longer happening, eventually the concentration of D end product will fall, and so then there will be less of this allosteric binding going on at A, and so A's active site will reform and the pathway, this enzymatic pathway, will resume. And so we can refer to this as a quick but short-term acting way of inhibiting a pathway to temporarily shut it down, but then resume activity when more end product is needed in the future. This illustration visually shows what the last slide was communicating in words. And I had spoken about moving from A to B to C to D. This simplifies it just a little bit, showing that intermediate A would be made as a first step after enzyme 1 acts on its initial substrate. And then there's a second enzyme in this pathway that will make an intermediate B. And then eventually an end product, we could call that C, will then be made as this enzymatic pathway runs forward from A to B to C. And as this end product is produced, it will eventually meet or even exceed the needs of the cell. And this negative feedback loop mechanism occurs when end product C travels up and makes contact with the allosteric site. The allosteric site is this area here where end product C will come and make contact. And that will effectively change the shape of the first enzyme of the pathway so that its active site no longer can bind to substrate, and then there will be no more manufacture of A or B or even C. And so end product will stop being made, and so long as the cell has met its needs, then this type of feedback inhibition will be in place. As the concentration of the end product falls, then what can happen is there will be less binding to the allosteric site on the first enzyme, and then this enzyme will resume its active shape, and this process will continue to flow to make more of intermediate A, B, and eventually end product C. And so this is how a cell can turn a certain synthetic pathway on or off through feedback inhibition. It is a temporary, quick, short-term way of using the enzymes like a light switch. And the most efficient enzyme to turn off is the first enzyme in the pathway. Rather than inhibiting enzyme number 2 or enzyme number 3, it makes the most sense to inhibit the first enzyme or the first worker. If this was an assembly line, you tell the first worker to take a break, and then the workers down the line will no longer have product to work on because the first worker stopped performing their task, and so the pathway shuts down.